Hello guys, this is me Dr. Kashan Kasit today here, and we are going to discuss today the topic which is HIV AIDS. HIV AIDS, since the identification of AIDS in 1981 and its causative organism which is HIV in 1983, 78 million people are estimated to have been infected with this infection, while out of that 39 million people have died, and at least there are 35 million people who are still living with HIV infection throughout the world. So I've decided to make a video on this critical topic of HIV AIDS. AIDS is a condition in which body's immunity is very low and it is prone to several other infections, and other infections can cause different clinical manifestations. So basically the AIDS is acquired immunodeficiency syndrome, which is caused by the virus which is HIV, human immunodeficiency virus, which belongs to the virus family which is retrovirus family. This HIV virus have two other strains, HIV-1 and HIV-2. HIV-1 strain is most frequently found throughout the world, while this HIV-2 is only confined to Africa and some other countries of the world. So basically these HIV-1 and HIV-2 are zoonotic infections, and their origin can be traced to a species we call chimpanzees, who are the natural reservoir of HIV and are most likely the source of this infection. Moving next, we are going to discuss the mode of transmission or route of transmission of this HIV infection. There are several ways by which this disease can be transmitted to other person. Number one here is the sexual intercourse, which is constituting about 75% of all the cases of HIV transmission. The frequency is more in male to male as compared to female to male. So this one is the first route of transmission of this disease. The second one here is the transmission via blood and blood products, which includes the syringes, needles and other appliances. So the blood and blood products involve the three categories of the people. Number one here is the intravenous drug abusers who are sharing the needles, syringes, etc. The second class here is the hemophilic patients who are actually receiving the large amount of blood from different multiple donors. The third one here is the patients who are infected with HIV blood. So these are the three categories of the people which are involved in this mode of transmission. The third one here is the perinatal transmission, which is the transmission of the infection from infected mother to the child during pregnancy. The fourth one is the occupational transmission. This is slightly less, but this occupational transmission can also occur in healthcare workers, laboratory workers and the person who are dealing with the disposal of HIV infected syringes, needle, etc. So the last one here is the transmission by other body fluids. Beside blood, there are the body fluids like saliva, tear, cerebrospinal fluid, pericardial fluid, seminal fluid, vaginal fluid, cervical fluid. All these are the fluids, and there is no definite evidence that HIV can be transmitted through these fluids. So these are the several modes of transmission of HIV infection. Next, we are going to discuss the pathogenesis of this disease. Now we are going to discuss the pathogenesis of HIV infection. The pathogenesis involves mainly the CD4 T helper cells, the reduction of which will result in the immunosuppression, due to which the body's immunity becomes very weak. So these are the steps through which virus get entry into the body, and it attacks the helper T cells and will result in the viremia. So let's start with the first step, that is selective tropism for CD4 molecule receptor. The HIV virus, which is having the glycoprotein GP120, this is GP120, have selective tropism for the cells which is having the CD4 receptor, like monocyte macrophages, CD4 helper T cells, epithelium of the cervix, Langerhans cells of the skin. All these have the CD4 receptors. So this GP120 glycoprotein of the HIV virus has selective tropism for all these cells. In the next step, there will be the internalization of, here is another protein which is GP41 glycoprotein of the HIV virus, which is occurring through the process we called internalization. In internalization, this is the GP120 of the HIV. This GP120 molecule combines with the CD4 receptors. These are the CD4 receptors present on the CD4 helper T cells. So this is actually this GP120 combines with these receptors with the presence of another CC4, which is cytokine co-receptor, which is necessary for the combination of GP120 with the CD4 molecule receptors. So once it is attached to this one, this GP41 is internalized into the host cell. So this is how the internalization of this virus GP41 occurs by this process. So after the internalization, there will be the third step, which is uncoating and viral DNA formation. So once the virus got entry into the cytoplasm of the host cell, then there will be the enzyme we called, here is the enzyme, this is the viral RNA, this is the enzyme we called reverse transcriptase. This reverse transcriptase will result in the formation of single-stranded DNA. This reverse transcriptase will convert this RNA into single-stranded DNA, and further there will be the another enzyme, DNA polymerase, which will convert this single-stranded DNA into the double-stranded DNA. So the double-stranded DNA formation occurs in the third step. So next, in the fourth step, viral integration occurs by another protein we call viral integrase protein, which will insert this double-stranded DNA into the nucleus of host cell, which is CD4 T helper cell. This is the host cell nucleus. So it is integrated into the host cell nucleus. So now the virus is present inside the nucleus of host cell. So in the next step, viral replication will occur, which is facilitated by certain cytokines we called Th1 cell cytokines, which are releasing interleukin 2 and TNF gamma. So these two are released by Th1 cells, and Th2 cells are releasing, liberating interleukin 4, 5, 6, 10. All these interleukin will help in the replication of this viral DNA. So the viral DNA is now getting the machinery of this host cell. Now it will separate to other parts of the cell. So the next one is the latent period and immune attack. During this latent phase, virus remains in there and it is not actually damaging the cell membrane. And during this period, the body's immune attack will occur, and body will fight against the virus by activating the CD4 and CD8 cells. So the body is now fighting against the virus by activating macrophages and by forming antibodies against the virus. But soon the virus will overcome this immune attack and it will damage the cell membrane of the host cell, and it will spread to the other cells as well. In the next step, CD4 T cell destruction occur, which I was discussing. CD4 T cell destruction occur, and the virus will move actually from this host cell by the process we called budding into the nearby cells. So now the virus is out of this CD4 cell. This is how it will destruct the T cell membrane and will result further in the viral dissemination, which we called viremia. So now the virus has destructed this host cell and it is now actually damaging the nearby CD4 T cells, and now it can spread to other part of the body as well. These virus particles are now spreading to the other part of the body, and they are actually now going into the lymph nodes as well, which are the reservoir of these virus now. And it is also spreading to other body parts by a circulation, and this will result in the viral dissemination and viremia. So this is how the viral dissemination occurs, and virus will infect the other CD4 cells. So this is all the pathogenesis of the HIV. Next we will discuss how this virus is actually presenting the symptoms in HIV patients, and also we will discuss there the management and treatment of this HIV AIDS. So hope you like my video. Thank you very much for listening. I'll see you in my next video.